So my name is Joann. I was diagnosed in November of 2021 with stage 4 non-small cell lung cancer, KRAS G12V.
My name is Mike Smith. I have stage 4 lung cancer. I was diagnosed in 2016, and much like most people's diagnosis, it came to very much stunning, particularly since I was a non-smoker.
I had several standard of care treatments, all of which resulted in progression. Luckily, I got onto a trial in February of this year. It was a targeted therapy, and it was a daily pill, which was wonderful, no more infusions. I had great quality of life for 6 months, very minimal side effects.
My initial diagnosis put me on the standard of care treatment, and that lasted 27 months for me. And that was about the efficacy rate, it was 24 to 26 months. So our thinking here was that if in fact I went on a clinical trial drug that was supposed to work as well or better, that I could perhaps extend my life. That's what led to my decision to go on that clinical trial drug.
So I definitely feel like my knowledge of the clinical trials and sort of my aggressiveness in the whole process certainly helped me.
I always tell patients like immunotherapy trials, like those patients that went on those trials that are alive today, 10 years later, it was a trial. It was as scary as the trials that we're talking about to now. No one knew that drug would work. And look what happened. That drug worked. It made the difference. It changed lives.
You know, it's such a blessing to be able to be anywhere after you get diagnosed with lung cancer. 6.5 years ago, I was diagnosed with non-small cell lung cancer and went through treatment that was chemo and surgery. And I had no evidence of disease and started getting connected with other lung cancer groups and started hearing about biomarkers and such and started pushing my doctor. Not getting the information I needed or I wanted, I pushed. And eventually, after about 2 years, ended up at a second opinion. The second opinion doctor is the one that told me I had KRAS. And I swore if I knew what it was, I wanted to connect with other people so that we can all learn what this means and how we can make it better to live with KRAS cancers. This is all about the hope that we've learned over the past couple of years. And I'm going to just be tickled to death to actually introduce you to our two speakers this evening. First up, we have, Dr. Estelamari Rodriguez is a hematologist oncologist at Sylvester Comprehensive Cancer Center, the University of Miami Health System, and she specializes in early detection and treatment of lung cancer. Okay, we also have with us Dr. Jason Porter, and you're a hematologist oncologist at the West Cancer Center Research Institute in Memphis, Tennessee. He's the director of the Lung Cancer Disease Research Group and specializes in treating patients with molecularly altered lung cancers and lung cancers with no actionable driver mutations. What are the most common types of biomarkers in the lung cancer, Dr. Porter?
So, a long time ago, we found EGFR as a biomarker, and then we have also the KRAS mutation. But we found KRAS actually a long time ago, and for such a long time couldn't target it. It can serve as number one, a biomarker that we can use to target for treatment, and then also a prognostic biomarker that kind of give us some information about how a tumor may behave. So we have both prognostic and diagnostic biomarkers outside of KRAS as well. So we also have PD-1 and PD-L1, which is a protein that we can study to kind of give us an idea about how our immune checkpoint therapies may work when we treat non-small cell lung cancer. And then we also have BRAF, and we have ALK and ROS1 and all of these different proteins that may be, as Dr. Rodriguez already alluded to, driving the cancer or just giving us information on how that different cancers may respond to treatment. What's really interesting is that we used to associate these biomarkers that were drivers with non-smoking. And now we have drivers that are actually associated with smoking. And so testing biomarkers is becoming very important for all patients, regardless of their smoking history. And so BRAF is one of those biomarkers that we can see present in smokers, as well as KRAS, which we talked about. And then MET exon 14 is another biomarker that's also an actionable biomarker that we can target in therapy that may be associated with smoking. And so we've now discovered that basically we need to do biomarker testing in all our lung cancer. And obviously we know that's very important. In order to even find out if a patient is going to be a candidate for that trial, we have to have their initial biomarker testing done. So not only have we increased the likelihood of them getting a relevant first-line therapy, but we also increase the likelihood that at the time of progression, we will have a good clinical trial that may be available for them. And so I think that getting back to biomarker testing, which ultimately can lead to access to clinical trials, and then making sure that patients are asking, what's my biomarker test say? Did you do my biomarker test? And we can't ask that enough. If you're at the clinic with your dad or your mom who's been diagnosed with lung cancer or any cancer, have you done any biomarker testing? It holds us accountable as providers so that we make sure that we're doing that for patients. And then we give them good first line of standard of care and good clinical trial options when a clinical trial is not appropriate in first line.
This whole message of treating patients earlier with chemo and the targeted therapy really got my attention, because we have seen in our practice patients that have this mutation, but we have to wait second line, meaning we have to go through chemotherapy and immunotherapy, and some of them get very sick, and it's very sad to me that they never got to see that targeted therapy. So being able to give them a response. So I think in the trial that combined carboplatin based chemotherapy with adagrasib, they saw responses like over of 60%. So they were able to bring up the responses that you wouldn't see with chemotherapy with the combination. So I think that patients kind of sometimes get a good chance to decrease the burden of disease, especially we know that some patients with KRAS mutations have a lot of burden of disease. It's very aggressive, and they have a lot of symptoms. So we're able to help them with all our drugs up front, and we can find a way to deal with the toxicity. I think we'll be able to help more patients.
Yeah, and earlier they're more likely to tolerate a more robust regimen, so it's also exciting to get to do that a little bit earlier. Definitely we suppress the subclones that are being driven by the driver that we're targeting, but then when we don't or aren't able to use chemo and or immune therapy up front, those other clones that grow through are resistant to our therapy and may even be more resistant to chemotherapy later. So we just see lower response rates. And then where we have these drivers in second line, I think, like you said, it's really sad to see those patients come to that point and then be clinically not able to tolerate whatever the therapy is. I definitely had a patient with a G12C mutation who wasn't a candidate for clinical trial in frontline. So when we got to second line, his effusion was refractory, even loculated and not amenable to easy drainage. And he was definitely not a candidate for the VATS where we go in and kind of clean that out. So trying to get him onto the G12C inhibitor at that point was, really, it wasn't possible. He couldn't tolerate it. And even though he was on for a very short period of time, he spent most of that time hospitalized and eventually went on hospice. But to imagine bringing that to an earlier line of his therapy, where when I first met him, he was walking into the clinic and really still robust, and being able to target at that time would have really been nice for him.
Yeah, it seems like we really need to be very strategic about what the treatments are. And if you don't have all this information going into it up front, you can't be strategic at all. You guys were mentioning, the EGFR group of patients is equal to the amount of KRAS G12C patients, right? And yet they've had a whole lot more studies, a lot more drugs that have been going on. And so we're not there yet when it comes to some of those other like KRAS subtypes, let alone some of the others as well. But I do keep hearing you both bring up about the smoking. Is it that important where it got here from? Or is that just like, it's just one of those things that keeps like getting reported out?
For me, it's not important where we got here from, but for me, what we have to remember is stigma. And we want to forget the actual stigma, but remember that stigma is a problem. And so the reason I always bring up smoking is because I was sitting in a room with a very, very smart oncologist who told me, well, this patient was a smoker, so they're less likely to have an oncogene driver. And I was like, well, 20 years ago, or 30 years ago, that would have been true. And so I like to use smokers and next generation sequencing and studies in the same sentence so that people start to make the association that you can definitely have a driver that's actionable, even with a history of smoking. And then again, the point that I made earlier about BRAF, MET-exon 14, and even KRAS, you're more likely to find those drivers with FDA-approved therapies in smokers. And so where it becomes relevant for me for a never smoker is that we know they're less likely to respond to immune therapy by itself. So, but for smokers, I like to just keep saying that and putting NGS testing in the same sentence. So people stop having this association that the oncogene driver patient is a young female Asian never smoker, because that's what we learned in medical school, And so to kind of really change that narrative, I don't want it to seem like I'm harping on smokers. I'm just harping on the fact that smokers can have drivers and that we have to study everybody. We have to.
Thank you for clarifying that, because that's exactly what I was hoping that you would say.
Right.
Okay.
Right.
Is that it used to be, oh, they're a smoker, don't need to test. Now it is a smoker. Okay, we definitely need to test. And part of the important part is what that means for what's next. And so that's why I'm really glad that you answered that for me.
We don't hear enough from people that have a good experience in a clinical trial. And I think that, we have a lot of patients that think that they're going to be experimented on and that they're going to be getting a placebo. And I work in an experimental therapeutic clinics where everybody gets a drug. We're not giving placebo drugs for those phase 1 trials, phase 2 trials. They're all about testing a drug. Now, we don't know how good those drugs would work, but as we look at the data coming out, and one of the things that has really changed in oncology is that we're getting results faster and drugs faster to patients, sometimes not as fast as you will need them, but much faster than it used to be. And I think biomarker testing has allowed us to do better trials, because now we can, find the right patients that are more likely to respond to some of these treatment options, when in the past, we will have a new drug and it will be given to everyone with or without chemo. And then obviously the responses were very low because we're not really looking for those patients that were more likely to benefit. And I would encourage patients, to really to think of second opinions as a good thing, not a bad thing. And I have patients that come to me and they're like, oh, I'm sorry, I'm going to go speak to so-and-so. And I'm like, I am delighted that you're going to go speak to someone else. First of all, because we're all friends, and no one here, if you have a doctor that gets upset about this, that's kind of odd, no? Because I think that if I was going through this, I would want to learn from other people, get a different perspective, and maybe another doctor can see something that was missed. And I think that it is good to do that, and you're not gonna, hurt your doctor's feelings, and they're not gonna stop treating you. And clinical trials the same way. Like the patients that are most likely to go into trials are patients that have been on trials before. So we have a lot of patients that we see that had a good experience, and that opens a whole new door of options for them. So I would encourage people to look. Fortunately, everything's consolidated in clinicaltrials.gov. Is a website where you can put in your mutation and your state and just get a readout of trials that are open. And then just knock on doors. All of those trials have an email of an investigator and a nurse coordinator. And I do that for my patients. I say, okay, we're going to write an email here and you're going to find out if this trial is open. And sometimes there's a trial right next door. that you hadn't even known about. And it's just, you have to do the work.
I think that an amazing resource for finding a clinical trial is your doctor. And that's I think where you need to start. But obviously you can't wait in case that it's not presented as an option for you. So if you have access, or if a patient has access to their biomarker analysis, or they know they have any particular mutation, you may just go and search that mutation clinical trials on the internet and see what comes up. And then you may even type in your city to see what comes up in your city. And so if your patient and your doctor is not really considering or giving you the option to discuss clinical trials, you may need to consider reaching out on your own. Definitely, we don't want to take it as a last-ditch effort, because number one, when we get ready to go on clinical trials, we have to control for as many variables as possible. And that means, are the kidneys functioning okay? Because if something happens on the trial and the kidneys weren't functioning well, it could be a complication of the kidney dysfunction. And then the drug kind of gets associated with that, unfortunate outcome. So we have to control for those things. So earlier in a disease like lung cancer, you're less likely to have some of those other disease issues that may exclude you from the clinical trial. So doing it earlier is better. And then when I thought back, because I came into my clinical practice around the time that immune therapy was starting to come into second line FDA approval. And I thought about the people who went on immune checkpoint inhibitor as an early part of those clinical trials before I was even in fellowship, and the ones who are still responding now from 2013, 2014, there are still some survivors. And so for those patients who went on early, the clinical trial sometimes gives you the best therapy before it's even available for general use. And so honestly, NCCN, which is one of our guideline kind of compendium, says that the best therapy for a patient with lung cancer or with any cancer is in a clinical trial. And if you end up getting that early, you have that long-term benefit. And then like 5 years later, the FDA is approving that therapy. And guess what? You got it way back when it was still, being discovered and understood. And so definitely not a last-ditch effort, and sometimes access to the most effective thing available.
So that's the beauty of this earlier phase trials, that they will take patients that have, and those are the patients that we want to offer treatment, patients that have failed immunotherapy, not failed because they didn't fail, the treatment didn't work. And then you want to find a different way to treat them. And so a lot of the trials that we do after immunotherapy is a new immunotherapy that is in a different way, or a vaccine trial, or a different mechanism to try to wake up the immune system. Because in a way, most tumors can possibly be treated with immunotherapy. They become what we call them hot tumors that are more likely to activate the immune system. And some tumors lose that capacity. Either you have an innate resistance that is not a tumor that is very hot in that end, and you can do something to it. Like we have tried different things like radiation followed by immunotherapy. There's a lot of exciting vaccine trials that are trying to kind of really push that immune response and what we have seen the success of the memory of the T cells in a longer way, infiltrating T lymphocytes, which are like white cells of your own body that get activated against the tumor and then given back to you to kind of do like a more directed immune therapy. So those are treatments that immunotherapy can possibly help patients with many different biomarkers, but still the biomarker driven research and treatment is very exciting, because sometimes you get those very deep and long responses. And one of the other excitement that I saw was really in the ROS1 and NTRK space. We had a new drug, repotrectinib. They all end the same way. But these drugs, like when we understand how you can bind these receptors and do better, then the next drug can have even a higher response. So some of these drugs have responses of like 70%, with like intracranial responses that are over 40%. So that means that we get better at the next generation of the drug. And that's what I hope for the KRAS G12C story, that the next generation of these drugs will be, we already have seen adagrasib and sotorasib have intracranial activity that is real. When I will give you an example from this week, I had a patient that I was getting ready for a next generation KRAS inhibitor, And I thought she was progressing, and I did all the studies to get her ready for the trial. And actually she had a complete response. So I couldn't put her in the trial. So I was all geared up to like, okay, we're going to neatly, quickly get you to this next KRAS inhibitor. And the one she was on was working very well. Like she had a response in the brain, and the pleural disease she had went away. So these are real things. And I think, again, we go back to identifying the biomarkers early so that you could get on the right track.
The other thing that patients can do, and that I think is really helpful, is that if you get a copy of your own biomarker testing, a lot of times right there on the result, it'll list out the clinical trials that are relevant for that particular biomarker, and even give you a listing of where those trials are. So a lot of times we assume patients don't want the information from the actual report from their next generation sequencing, but as we educate and kind of really get advocates out there, letting patients know how important the biomarker testing is, they can find often a list of the relevant clinical trials for their biomarker right there on the report. And that can kind of guide them in their initial searches for clinical trials for their biomarker.
And I think we can't forget that crowdsourcing for clinical trials is really one of the most effective ways. So I have patients that have found trials because they were part of a group, of a patient support group, and someone in another state was in this trial, and that kind of opened up an opportunity to really look at a trial that was in their same state. So I had a patient that came from Jacksonville, which is pretty far from us, but she found the trial through the patient support group. And you can reach out, and people that have gone through other trials already have made connections, and that can be very helpful.
I think a lot of patients want to know what's the purpose of a trial, and are there any kind of risk associated with being in a trial? And I will answer the risk question first. And it's a little bit kind of counterintuitive, but if you think about it, in a clinical trial, you're monitored so closely, and any risk that would be associated with any therapy are much more likely to be discovered while you're on a clinical trial. So if there's going to be any trouble with your kidneys or your liver while you're on a clinical trial, which could happen with the chemotherapy that's already approved anyway, you're going to be getting labs more frequently. You're going to be coming in to see the nurse a little bit more frequently, and possibly even just phone calls to say, hey, how's everything going? Are you having any issues? From the clinical trial coordinators. Our coordinators do that sometimes. And so I think you're monitored a little bit more closely. And so I feel like diminishes risk that may be associated with the clinical trial. And as we get to the different stages or phases of the clinical trial, we have more experience with that particular drug or that molecule. And so there's even more information about what risk may be out there. So there's a phase 1 clinical trial that Dr. Rodriguez mentioned that she helps to facilitate at her clinic. And so that is pretty much the first times that these drugs are being used in a single agent, or either in combination with another chemotherapy or agent. And then in the phase 2 clinical trials, we have already established that it's effective, or that we can see responses. So we want to study a bigger population, and we're seeing more patients now being exposed to the medication to confirm that it works. And then in phase 3, we're pretty much doing a much larger trial where we're comparing it to what's already considered to be the standard of care. So this is the way we treat this, but now we have this new option, and let's compare the two to see if one is better. Oh, it's better. And now we have a new standard of care. So that's kind of like the phases of clinical trials. But I feel like you get monitored so closely across that whole spectrum that it kind of reduces risks that you're going to have complications. And at least if you're going to have them, they get identified much earlier.
What are the typical eligibility criteria? My mom can't do trials near her because of her poor ECOG PS, and she's on oxygen. You guys give us some guidance, Dr. Rodriguez?
So that gets to the point that we need to develop trials for the patients that are in the real world, and that is a real world patient. So patients with lung cancer, some of them, because of their disease, they're going to be on oxygen, and patients with brain metastases are going to need clinical trials, and many trials have excluded them in the past. So we need to encourage more trials that are for patients that need them and have a lot of symptoms. Now, sometimes there are patients that are too sick to go into a trial because they're new drugs, and we really don't know how they tolerate them, or we have serious concerns about toxicity, and you kind of have to trust your doctor that those eligibility criteria are there to make it safe. Although we do know that sometimes it's to make it too safe, they're excluding patients that need treatment. And there is a big movement by the NCI to make trials for real world patients. Don't be afraid to ask about clinical trials, because if you have a diagnosis of lung cancer where a lot of patients are going to progress, this is the fight of your life. And if you're not open to clinical trials, you're kind of closing your eyes to opportunities I always tell patients, like immunotherapy trials, like those patients that went on those trials that are alive today, 10 years later. It was a trial. It was as scary as the trials that we're talking about to now. No one knew that drug would work. And look what happened. That drug worked. It made the difference. It changed lives. So I just want to give people the sense of hope, that clinical trials offer hope, that people who do clinical trials do get better outcomes because they're monitored very closely for risk. And it just really gives you an opportunity to do better and really kind of push the envelope in a disease that can be relentless.